This is a production of Cornell University.
Today I'm going to talk about one of the chapters of my dissertation, and the title of this talk is Broad Spectrum Resistance to Hemp Powdery Mildew at No Cost. And we hopefully will answer that question by the end of this presentation. So we've heard about hemp before, but I'm going to introduce and here I can change the pointer. Perfect. Okay, so I'm going to introduce the disease, which is hemp powdery mildew. It's primarily caused by an ascomycet fungi, which is called Golobinomyces ambrosia. Important here is that it's an obligate biotrophic fungi, and that means that it grows on tissue that is alive. As you can see on the panel A, it appears as white patches, and on very susceptible genotypes, those patches will expand and coalesce and cover the upper part of the leaves. This is a really big problem for high cannabinoid hemp cultivation, especially the cultivation indoors, and it will reduce the quality of those commercial high cannabinoid market products. So it's a really big problem. It's also a polycyclic disease, which it means that there are multiple cycles of new infections within one cycle of the crop, which makes it difficult to control. I'm also going to introduce genes that we're working with, which are mildew locus O or mildew resistance locus O, which are highly conserved plant protein family, a highly conserved plant protein family specific to plants. There are 7 clades for these genes, and more or less the clades are involved in different biological processes. The proteins that they encode have 7 transmembrane domains, with the N-terminal part on the outside and the C-terminal part on the inside, which also has a calmodulin binding domain. And until recently, we didn't know what they did, what their biological function was, and they have been around or have been discovered a really long time. So in barley in the 1940s. And we now know that they work as calcium channels, which makes a lot of sense. We also know that they are also associated with resistance or lack of susceptibility to hemp powdery mildews. And this has been studied in other plant species. For some plant species, there are no negative effects of mutating these genes. So these work recessively, so you have to mutate the 2 copies of a gene, or all the genes that are in the clade 5 for these genes, for the cotyledonous plants. In monocotyledonous plants, the clade 4 MLOs are the ones that are associated to this lack of susceptibility to powdery mildew. So in other plant species, we see that I included pictures for the 2 cases for cucumber and for Arabidopsis thaliana. Both of them have 15 MLO genes, and they have 3 that are in clade 5. For cucumber, in the case of cucumber, when you mutate the 3 clade 5 MLO genes, you see that the plants are shorter and they have smaller leaves, and also the fruits are smaller. And in the case of Arabidopsis, if you mutate all of them, you start seeing some premature senescence, some spontaneous callose deposition, also the reactive oxygen species get deregulated and you have premature cell death. So if you're thinking about incorporating these mutations to confer a lack of susceptibility to powdery mildews in your crop, you don't want these effects. You want to check that this is not going to happen, especially from a yield perspective. Also, Kunsch and Bastruga made this figure years ago, and they are representing the type of MLO with the powdery mildew susceptibility and possible pleiotropic phenotypes. So for wild type plants, you would be susceptible to the powdery mildew without any resistance genes playing here, but you wouldn't have any negative pleiotropic effects. If you are working with a strong MLO mutant, on the other hand, you won't be susceptible to the powdery mildew, but you might start seeing some negative effects. So what do we know about this in cannabis? There are 15 predicted MLO genes in the genome of cannabis sativa. This is from a paper from 2021. And 2 of them belong to clade 5, MLO1 and MLO4. We also know that, and this is published results, and this is from our lab, that in a QTL mapping study, when a resistant parent was crossed with a susceptible parent, and those F2s were analyzed, that there found a big peak on chromosome 1, and that was around 20 megabases into that chromosome. We also had the genomes for those 2 parents, and they saw that there was a big insertion, around 6.8 kb insertion between exons 10 and 11 of that gene, and that was probably conferring this lack of susceptibility to hemp powdery mildew. So with this information, the hypothesis that I'm working on, where if you're homozygous for this allele, you're going to be resistant or not susceptible to hemp powdery mildew in other genetic backgrounds. And I'm thinking about this because the original plant, FL58, is a clonal cultivar, which means that we're not looking at how this allele is segregating in a population. And we don't know what happens in other plants that were not bred for high cannabinoid, as FL58 was. The second hypothesis was that if you're homozygous for this allele, you're not going to see those other negative effects that we see in other plant species. And this is because in FL58, we are not seeing them, but we want to check that it won't happen if we start trying to introgress this allele into other plants that are very different from FL58. So how did I go about this? We had 3 donor parents for this allele, and we had other diverse parents that represent genetic diversity in the germplasm that was available to us when I started. We crossed those parents, we generated F1s, we crossed 2 F1s, and we generated F2s that would be segregating 1 to 2 to 1 for this allele. This is a complete crossing scheme. The 3 parents, one was resistant to hemp powdery mildew and homozygous for the allele, and the other 2 ones were heterozygous, moderately susceptible, and highly susceptible. But all of them were bred for the high cannabinoid market class. Then the diverse parents, we had XX females, XX monoecious plants, XY plants, plants that were bred for grain purposes, fiber, high cannabinoid, and even one was a feral collection. So we're trying to capture that genetic diversity with this diverse lines. We planted this F2s in a randomized complete block design with 4 blocks in a field here in Geneva. I had the 10 parents, the 3 donor parents, and the 7 diverse parents, 3 by 7. Those 21 families were in the field last summer. Had 12 plants per parent and 60 plants per F2 family. What traits did we measure? The most important one was the powdery mildew disease severity, but we also measured flowering time, sex, height, wet biomass, and other architectural traits. I included representation. It's a figure from Carlson from et al. From 2021, and you can see how those measurements would look like in a plant. We also collected leaves. We extracted tissue. We extracted the DNA, and we run our pace markers to see what the genotype was for each of the plants. And what did we see? Sorry, we inoculated the field twice this summer because it was super dry and it wasn't. We were not having the best conditions, most optimal conditions for the development of this disease. And I rated 5 times during the course of the summer. So here we go. We saw that the homozygous MLO mutated plants were resistant to hemp powdery mildew. That's awesome. We calculated the area and the disease progress curve. If you worked with diseases before, you've heard about this, but this is calculated. It's a measure of the disease progression over time. So with those 5 ratings, I calculate that area under the disease progress curve. And this is what we are seeing in our y-axis over here. We see the genotype of the plants in the x-axis, the 7 diverse parents on the upper part, and the 3 common parents or donor parents on the right side. And let's look at what happens in Candida number 2, which was from the diverse parents, the most susceptible one. When you cross very susceptible plants with resistant plants, that's when you see the highest difference between the groups. So our homozygous MLO1 plants were resistant to hemp powdery mildew, and the heterozygous plants and the homozygous wild-type plants, all of them had different levels of disease, and they were susceptible to hemp powdery mildew. You see that in other cases, we don't see differences between the groups, and we think that that's because we might have crossed 2 F1s that were on the most susceptible, most the resistant side. So if you cross 2 plants that are not so susceptible, you are probably not going to see that many differences in the F2s, and they are going to be on the more resistant side. Also, it was a very dry summer, so probably in other conditions, we might have seen like a earlier development of the disease that translates to higher AUDPC values, and that would separate those groups a bit more. Also, the second hypothesis was about negative pleiotropic effects. Did we see some? And the answer is not really. I'm showing you kind of heat map, kind of. We see the 21 F2 families on the y-axis, the different traits that I mentioned at the beginning that we rated. And we also see the direction in which those differences went. So if we take the average for each of the genotypic groups for each of the traits, you see that for some of them, there are higher or greater values for the group that was homozygous for the mutated MLO1. And there are other cases in which the homozygous wild types had greater values. So that it's going both ways. I also added asterisks to show the ones that there were significant differences, because seeing differences doesn't mean that they are significant. I'm going to guide you and concentrate on the flowering time trait, because this is a really important one, and it's the one that has the most significant differences too. So flowering time. We know that, or let's look at this circus plot with the chromosomes for cannabis sativa. This is from 2023. And let's look at chromosome 1. That's where the MLO1 gene is, and it's at the beginning of the chromosome 1. We see, or we know already, that there are multiple flowering time genes around that area. So they are probably linked in coupling phase or in repulsion with the different alleles that we are working with and the different genotypes. And that is probably going to affect the flowering time of the flowering time of the plants. We also know that plants that flower earlier have less time to get all the nutrients, to photosynthesize, to grow vegetatively. And therefore, they are going to have a shorter stature. They're going to accumulate less biomass and have thinner stems. So if we see significant differences in flowering time, it's not weird, or might be expected to see differences in the other traits that we measured. So I hopefully convince you of that. And the take-home messages are that if you're homozygous for this mutated allele, you're going to be resistant to hemp powdery mildew. That is answering or testing our first hypothesis. And then for the second hypothesis, that was about the negative effects, we see that there's linkage between our MLO, the MLO, different MLOs alleles and flowering time genes. So if you're introgressing this, as we did just with crosses, into elite backgrounds, you should pay attention to those other flowering time genes that are linked with the allele. The allele itself does not apparently per se produce deleterious effects, at least in the populations that we were testing. And there are no other negative deleterious allele or death gene that is linked in coupling phase with the alleles, the different sources of alleles that we were using in this study. So with that, I want to thank the members of the Smart Lab, because I've shown some pictures and it did not work alone. Had a lot of help, so thank you. And mentioned some of the funding sources. I had the Fulbright scholarship for the first 2 years, and then we had money from New York State Department of Ag and Markets, FFIR, and my EOA-ship. So thank you, and I will take questions.
All right, thank you, Lou. Any questions from the audience? All right, I guess I'll go. So, Lou, you said in the beginning that you have many different diverse kind of parents that you're including in your overall population coming from many different areas. Did you see any kind of like the fiber ones versus some of the other traits or other kind of commercial areas were doing better or worse compared with crossing two specific donor lines?
So I for the negative effects, is that for the negative effects parts?
Yeah, like more likely, maybe if fiber plants were more likely to have negative effects or like less likely or something like that.
Well, I'm still looking at those results because there are some families or parents that look like have the most significant differences. So those are Anka, Bielobrzeski, and some crosses with Logan. They also have those differences with flowering time, so that might just be associated when they flower, and that's why we're seeing the differences. We know also that for flowering time, the only plant that flowers really late there had significant differences in the opposite direction of all the other crosses. But yeah, it's difficult to generalize. I think it's very family specific.
Interesting, thank you. Any other questions from the audience? Oh, Jean-Luc?
Can you go back to the slide with the sort of heat map slide? It looked like recessive MLO generally causes later flowering, right? That's more frequent on that left-hand column. And then the other columns have to do with growth. And you point out later on in your talk that the later you flower, the more you're going to grow. And the other columns, I would say, are the ones that are really mixed. So I wonder if you tried to analyze sort of some of the growth traits per growing degree day before flowering or something like this. In other words, you try to adjust for the flowering time to see rates of growth pre-flowering. Might be a mess, but anyway. I mean, I don't know. When I look at this, it looks to me like recessive MLO is a bit negative. I mean, later flowering generally is bad. And okay, comment.
No, I had not thought about doing that. I'm wondering if I would need like traits over time, so I don't have height every week, for example. Or if I could do that.
The final value. I don't think you need traits over time.
I will look into that. That's super interesting. I hadn't thought about it. Yeah, and about the flowering time, yes. Flowering time in cannabis, some people or in some latitudes, you prefer plants to flower earlier so that you can harvest them earlier too, before the first frost and before there's snow in the ground. But for some more southern latitudes or closer to the equator, you would prefer plants to flower later or have a delayed flowering. Or if you can control it in your greenhouse, you're not so subject to what's happening outside. But yeah, thank you. I am going to write that down. And if you have another way of representing these results, I was playing with it so many ways. I just landed on this one, but I'm open to analyzing it or representing it in a different way.
All right, let's give a hand for Lou.
Thank you.
Thank you, Lou.
This has been a production of Cornell University, on the web at cornell.edu.